The next part we'll talk about is the fovea, this being in the retina. It's the central point, the directly back, the very straight back onto your retina is the fovea, where the cones, the eye's cones are clustered. So because of this, there's little color vision in the farthest periphery of our vision. And we'll do something in class next time we see each other to notice this and to show that we don't really see color in our periphery. So let's look at this picture here really quickly of the eye. So this is an eye as if it were looking up. So as the light comes in through the pupil and to the lens, focuses on the retina, the fovea is directly back from the middle of the eye. This is a zoomed in version of the fovea. Notice how it's a little bump. It bumps in here, just like it bumps there in the eye. Now, all these little green things here are cones. Cones are what allow us to focus. Cones are what allow us to see color. Write that down. Cones, color, and they allow us to focus. That's why we have cones in our fovea. Notice how it says up here that it is a rod-free area. There are no rods in our fovea. So the purple on the outer part, outside of the fovea, those are rods. We do have some cones out there, but the farther we get away from the fovea, the fewer cones we have. We will test this, that we can't see color in our periphery when we see each other again. So let's talk a little bit more about this rods and cones. There's specific information over here, but you can go ahead and pause and write these things down, but I'm going to talk about them as well. Notice that there are way more rods than there are cones. And again, we talked about the location. The cones are in the center. They're in our fovea. The rods are in our periphery. Cones allow us to see color. They are color sensitive, where our rods are not. Rods do not see color. So sensitivity in dim light, cones are not. Because again, we have to have a lot of light in order to focus on something. Okay, if you need to prove this, just turn off the lights really quick and see how difficult it is to see in the dark. So our rods are really, really useful when it's dark because they allow us to see in dim light. You should write that down. Rods allow us to see in dim light. So ability to focus and detect detail is through our cones. Our cones allow us to see the detail because they're... Now, number of bipolar cells. This is crucial. Cones each have their own bipolar cell. And we'll see a picture of what that looks like in a minute. Whereas rods share a bipolar cell with other rods. So let's take a look at this. So bipolar cells receive messages from our photoreceptors, okay? And this is a zoomed in picture. So the yellow here is a cone. Notice it has its own bipolar cell, okay? It kind of shows that other rods come in. That's not what it's showing though. The cone has its own bipolar cell. Now all these four rods though, they have to share this one bipolar cell. So think of it as if you are the only car on the highway. You are the only cone for your bipolar cell. You're the only car on the highway. You can get there fast to deliver your message, right? And it's not just being about fast, is that you're very clearly sent, you could say. Your message is very clearly sent. Again, sending a very clear message, allowing you to focus and see color. But if there's a lot of you, a lot of cars on the highway, if there's a lot of rods on the bipolar cell, you're not going to get there as quickly and your message is not going to be as clear. So remember, cones have their own bipolar cell, rods have multiple ones. And then that message from the bipolar cell goes to the ganglion, which then the axons of ganglion cells create the optic nerve. Feature detection. So we have nerve cells in our visual cortex that respond to specific features. So we have specific cells back here that are strictly in charge of allowing us to see edges, angles, length, and movement. One for each of those, multiple ones for each of those, but they only focus on edges alone, angles alone, length alone, those kinds of things. So there are even some feature detectors that are specifically sensitive to the human face. It's what allows us to notice that one face looks familiar to us, that one face looks different from another face, even though everybody has a nose and has eyes and cheekbones and hair. It allows us to notice that that is a different face, a different human being than another person. Visual information processing several aspects of a stimulus simultaneously at once. It's called parallel processing. So the brain divides a visual scene into subdivisions like color, depth, form, and movement, and processes all of those things at the same time. It's like when your computer is downloading 4 different things all at the same time on your screen. This shows the process of how we're getting information. So here's the scene, right? You looking at this picture of this gentleman. Retinal processing, so our rods and cones see it, goes to bipolar, goes to ganglion. So then the information is sent to the visual cortex. And I actually should put something in here. It goes to your thalamus first, and then it goes to your visual cortex. And that's where we have feature detection. So the brain detects things. They have specific cells responding to these things. So from the visual cortex, it's sent to other association areas where it's more about abstract. The brain's higher level cells respond to combined information from our feature detectors, and then from association areas to memory. The brain matches the constructed image with stored images and allows us to recognize something. A very big, long process that takes a split second. So here, until the end of our notes, we're going to talk about theories on color vision and why or how we're able to see color. Trichromatic theory, tri being 3. It's based on behavioral experiences from Helmholtz, who suggested the retina contains 3 receptors, sensitive to red, some sensitive to blue, some sensitive to green colors. So we have each different color for specific receptors. So in research about vision, they have found that different colors make different kinds of waves, and we have receptors for those 3 colors. So let's talk about the opposite of that, or when people might have difficulties with that, in color deficiency or blindness. And when I say blindness, I don't want you to think that people completely don't see color at all. In fact, completely only seeing black and white is extremely rare. So normally when people are colorblind, they just simply have deficiencies. It is a genetic disorder that prevents an individual from discriminating between certain colors due to a weakness in or lack of one of the cones. The most common form of color blindness or deficiency is difficulty distinguishing between red and green. Again, complete color blindness does exist, but it's very rare to only see in black and white. So this is a colorblind test, and I actually have had a student before who found out he was colorblind because of this test. Each of these round circles here has a number in it, and it's kind of cheating for you here because it tells you what the number is. If you cannot see the number in amongst these colors, you may have a color deficiency. So this is what the world looks like to someone who is colorblind. So normal vision is on the left here with the stoplight and with the purple grapes. But then this is what it looks like on the right, the stoplight, and they don't look so purple. If someone has a red-green deficiency, so normal vision is over here on the left again, and then the red-green deficiency on the right. You can find more pictures like this at critiquewall.com. Some more examples: the non-color deficiency up top, someone with color deficiency and what they see down here on the bottom, and that is just, you can see the images, it just lacks the same color. Now, another theory, one equally important, is opponent process theory. Opponent process theory. So Herring proposed that we process 4 primary colors opposed in pairs. And these pairings are red and green, opposite on the color wheel, blue and yellow, and black and white. So the cones are red, green, and blue, but then the retinal ganglion cells, we can receive and get a combination to receive all 6 of these. So after images is what helps us see this. So what I want you to do here is see this little dot right there. Gaze at the middle of this for about 30 seconds. When it disappears, stare at the dot and report what you see. So I'm going to have you pause and look at this for 30 seconds. Go ahead and pause and then play when you're done. Do you see an American flag? And really all you have to do is look at the white, like over here, the white. Now let's do this again. Gaze at the middle of the light bulb for about 30 seconds. When it disappears, or what I should say is just look over to the white and tell me what you see. Now, color constancy. The color of an object will remain roughly constant even if the lighting and wavelengths change. So what this is simply saying that color is constant. It does fluctuate slightly, but it's still the same color.